All right, the last installment for Chapter 1, which is very important, are the microscopes. There are always questions about the microscope, so let's review these. The light and electron microscopes, they like to trick you up as far as which one would be best to do this. So let's talk about magnification and resolution for just a moment. On the left, we have a picture of the, I believe these are antelope? No. I don't know. Ibexes, maybe. The act or process of enlarging the physical appearance or image of something. So we've magnified it. This is what you would see with regular human vision. Magnify it 7 times or 10 times, perhaps through a camera. Resolution is something a little bit different, and it's the ability of a microscope or a camera to distinguish between 2 points. Magnification without good resolution and an image will appear blurry. This is usually the difference between really expensive microscopes and really cheap microscopes. The really cheap ones may say they have a magnification of 100 times or 400 times or whatever it may be, but the resolution involved with those lenses may be the difference between cheap and expensive. Magnifying glasses, also called hand lenses. You guys are good with those? Let's talk about the compound light microscope. This is what we have in class and what we've been using this year. And it's a microscope that uses light to illuminate the specimen that is magnified by 2 lenses. And the 2 lenses, you have one here in the eyepiece and one here that you can change. The facts that you need to know about the compound light microscope is that it magnifies up, ours magnify up to about 1,000 times. Some can do up to 2,000 times. It won't do any more than 2,000 times though, and that's a good number to remember. Because if it's more than that, then you know that it has to be an electron microscope. These are good for viewing cell shapes and perhaps to see a nucleus in a large cell. You really don't see very many organelles with a compound light microscope. Here's a picture of bacteria cells and yeast cells under a compound light microscope. You can see a little bit of stuff going on in there, but not a whole lot. Let's talk about the electron microscope. Electrons are beamed at a specimen and a 3D image can be seen. These are high-tech microscopes usually found in labs and on college campuses. They can magnify up to 10 million times. They're good for viewing cell organelles, viruses, and other smaller than cell objects. So if a question asks you anything about something inside the cell, or a virus, or a particular molecule, you're going to need an electron microscope. There are 2 types, the scanning electron microscope and I can't remember the other one off the top of my head. Either way, both of them do a similar thing. Here's the picture of the yeast cells that we saw in the last slide with an electron microscope. We can actually see inside the cells and we can pick out some of the shapes. This is that E. coli bacteria that was on the last slide, and we can actually see some of the flagella and cilia that's on this E. coli bacteria with the electron microscope. One thing that you also need to know about the electron microscope, in order to use it, the specimen must be dead. There is no way to look at a live E. coli bacteria with an electron microscope. This idea of shooting the electrons through it means that it is most certainly dead. Dissecting scopes. These are stereo, meaning they're in 3D because they have 2 eyepieces. Having 2 eyes is what enables you to see in 3D. So the eyepieces here enable you to see in 3D for whatever you put on the stage. The magnification is generally less than 100. Another thing to know, the light source can come from up here, so you can shine a light down on top of something if you were looking at, say, the facial features of flies, or if you were looking at the delicate wing pattern of a butterfly. You might use a dissecting scope. The nice thing about this is that the organism can be alive, and that's pretty cool. So, in retrospect, if you're looking at cells or the shape of cells, you're going to use a compound light microscope. So looking at tissues, things like that. The dissecting scope is going to be looking at whole insects. So if you want to look at the pattern on the face of a honeybee, they tend to like to do that, or the pattern on a wing of an insect. Something that's small, but not necessarily microscopic. You would use a dissecting scope. Anything that's smaller than a cell or inside a cell, you're going to use the electron microscope. Here are some pictures of these in use. You see how he has a light coming from above. He's looking through here, and he's got a little light source right here shining on it. And here's, you can kind of pick through some very fine things, pick out different grains of sand if you wanted to with a dissecting scope. You do need to know some basic SI units. This is the metric system. So your grams, kilograms, liters, meters, all of those types of things. And that's it.